Cleavage. Cleavage is the division of cells in the early embryo. These divisions result in cells called blastomeres. Once a zygote is formed, the zygote is unicellular one which undergoes multiple division to form what is known as the multicellular organism. And this process of division of this unicellular cell into multicellular organism, the division of cells is called cleavage. These divisions result in cells called blastomeres. So the unicellular organism, when it undergoes division, it forms cells, and these cells, by the multiplication, how it is formed, these cells are called as blastomeres. And these blastomeres, actually they occupy different regions and differentiate into several types of body cells that forms a multicellular organism. Cleavage is a process of rapid and synchronous mitotic cell divisions of the zygote to form numerous cells for building up the offspring's body. And cleavage, it is different from other forms of cell division in that it increases the number of cells without increasing the mass. So there is no growth phase. There is no growth phase in this mitotic division. Only the synthesis part is taking time along with that mitotic phase. And cleavage ends with the formation of blastula. Once the blastula stage is formed, then the process of cleavage ends. So the first division of zygote up to the stage of blastula is what is known as cleavage. As the cleavage progresses, the resultant daughter cells, which are called blastomeres, actually they get reduced in size because there is no growth that is taking place. Only the single cell, the single zygote, that is dividing into numerous blastomeres without change in size. The zygote, what size the zygote has when it reaches the process called blastula, up to that stage there is no differentiation in, or there is no change in size of this zygote, because during cleavage there is no growth in the blastomeres. The total size and volume of the embryo remains the same. The cleavages result in a compact mass of blastomeres, and these blastomeres, the zygote now becomes known as morula. It is called morula. Morula is the zygotic stage or the embryonic stage where cleavage occurs, and you get a compact mass of blastomeres. This gets transformed into blastula. While the wall of the blastula is called the blastoderm, the central cavity of blastula is called the blastocyst. So this is how a cleavage takes place. A unicellular zygote that divides. So that single cell that divides into 2. Then it divides into 4, 8, and the process goes on. But there is no increase or difference in size. The only thing is the blastomeres are getting reduced. But there is no change. There is no change for this growing embryo. This growing embryo, growing in the sense we say it is growing because from unicellular it has become a 16 to 32 celled zygote. So this stage, this is called morula, where you have a 32 celled zygote. That stage is called morula. The cell size decreases, but the total mass and the volume remains the same. And the characteristics of cleavage, all the division of the zygote are mitotic, but this mitotic stage that takes only 30 minutes. Interphase in cleavage division is very short and does not involve of growth, so that the resulting blastomeres become smaller in size as their number increases. The total cytoplasmic mass that remains the same as there is no growth phase. And cleavage division clearly differs from ordinary mitotic division, in which For mitotic division, the daughter cells, once division takes place, the daughter cells grow, and when they reach the size of the parent cell, then again division takes place. But here it is not in such a manner. Once division takes place, immediately the second division takes place. It just takes about 30. So this is the somatic cell cycle, where you have a mitotic phase, the S phase, the growth 1 phase and the growth 2 phase. Here what happens in this interphase stage, only the DNA synthesis or the S phase is present in cleavage cells, as well as the mitotic phase. So the time that is required is less. It takes just 30 minutes. Somatic cell cycle, it contains 4 phases: G1S, G2 and M. The cleavage cell cycle, it comprises only 2 phases, the M and S. M and S phases of cleavage cell cycle, they are shorter than those of somatic cell cycle. The ratio of nuclear mass to cytoplasmic mass is very low for the zygote, and during cleavage nuclear material increases because new nucleus is formed for each blastomere. As a result, the nuclear cytoplasmic ratio, that again enhances. This makes a cell more active. So this is a normal somatic cycle, and this is a cleavage cycle which takes just 30 minutes to complete, and after one division, immediately the next division. Shape of the embryo does not change during cleavage because there is no movement of blastomeres. It remains as such, rounded ball-like structure. Early cleavage is regular and it is synchronous, that is, all the blastomeres divides simultaneously. With the result, number of blastomeres doubles always. It becomes 2, 4, 8, 16, 32, 64 at each cleavage. And cleavage becomes irregular so that the blastomeres no longer double at regular interphase after that stage. DNA synthesis occurs faster during cleavage to facilitate duplication of chromosomes for rapid division. This is how doubling takes place. A single plane that cuts it into 2 cells, then again a cut that divides it into 4, again one equatorial cut that divides it into 8. So this is how the top of the cell looks like. Cleavage, besides producing a large number of cells by rapid division, also segregates different substances present in the cytoplasm into different cells. What was present within that unicellular zygote, that is being differentiated into different cells when the cell number increases, and this is actually the maternal mRNA is being used for this contribution. And consumption of oxygen, that greatly increases during cleavage to provide, because metabolically it is active, it requires energy, and for this process consumption of oxygen greatly increases. The blastomeres remain adhered together during cleavage. The protein synthesis that occurs in cleavage is directed by maternal mRNA contributed by egg. And the planes of cleavage. There are different planes in a cleavage that divides the egg in, or the unicellular embryo into multicellular morula. Depending on the position of the cleavage furrow, the planes are named. The first one, that's a meridional plane. For the growing embryo, you have an animal pole as well as a vegetal pole. The plane that cuts the animal pole and vegetal pole, that passes through the animal pole as well as the vegetal pole, that is known as meridional plane, and this is a central one that cuts the zygote into 2 equal halves, and this one is known as meridional plane. The plane of cleavage lies on the animal vegetal axis. It bisects both the poles of the egg, and the egg is divided into 2 equal halves. And vertical plane, that's on either side of the meridional plane. If you cut, then that is if a plane passes through either side of the meridional plane, then that is known as vertical plane. The furrows again, they pass through the animal vegetal axis, and the cleaved cells may be unequal in size because meridional it's passing through center, whereas vertical it is through sides. So the cells may become unequal in. And equatorial one, this cleavage plane bisects the egg at right angles to the main meridional plane. It lies on the equatorial plane. It divides the egg into two. And the latitudinal plane, it's just like the vertical plane, where if a vertical plane is on either side of the meridional one, the latitudinal plane is on either side of equatorial plane. Again, this results in unequal size of blastomeres, and it is also called as transverse or horizontal cleavage. And types of cleavage on the basis of potentiality, how it develops. It is differentiated into indeterminate cleavage and determinate cleavage. Indeterminate cleavage, it divides into two. The cells are separated from each other. Each divides into two different organisms. Whereas determinate cleavage, if you separate this, then the cells die. And the cases of determinate cleavage, also called mosaic cleavage, is in most protostomes. It results in the developmental fate of the cells being set early in the embryo development. Each blastomere produced by early embryonic cleavage does not have the capacity to develop into a complete embryo if it is separated. It is determinated. That is why it is known as determinate cleavage. It is indeterminate. It is characteristic of deuterostomes. The original cells in a deuterostome embryo divides. The two resulting cells can be separated, and each one can individually develop into a whole organism. Types of cleavage on the basis of amount and distribution of yolk, and this could be classified into two: holoblastic as well as meroblastic. Holoblastic, you have radial, bilateral, rotational and spiral cleavage, and meroblastic, it is of two types: discoidal and superficial cleavage. Total or holoblastic cleavage. In this type, the cleavage furrow, it bisects the entire egg. Such a cleavage may be either equal or usually holoblastic cleavage, it takes place where the yolk content is less. Equal holoblastic cleavage in microlecithal and isolecithal eggs. Cleavage leads to the formation of blastomeres of equal size. Example is Amphioxus and placental mammals. Unequal holoblastic cleavage in mesolecithal and telolecithal eggs. Cleavage leads to the formation of blastomeres of unequal size. Among the blastomeres, there are many small-sized micromeres and a few large-sized macromeres. These few large-sized macromeres are because the yolk content is present, and when yolk content is present, the division takes time for the division to complete into two. So what happens is those cells become larger in size and they form what is known as macromeres. Meroblastic cleavage. In this type, the cleavage furrows are restricted to the active cytoplasm found either in the animal pole, that is macrolecithal egg, or superficially surrounding the egg, which is called centrolecithal egg. Meroblastic cleavage, they may be of two types. Discoidal cleavage. In macrolecithal eggs, plenty of yolk is present. The cytoplasm may be restricted to a narrow strip within the animal pole. Hence, cleavage furrows can only be formed in this disc-like animal pole. The entire egg is not involved in cleavage. Such a cleavage is called discoidal meroblastic cleavage. And example is birds and reptiles. You know, chick egg, it contains yolk, and this yolk actually takes time for this to divide because the fatty acids are more. So what happens is they form larger mass. They form larger mass, and division is slow. And actually division takes place only on top, where you have a disc-like animal pole. So this forms discoidal meroblastic cleavage. Type of cleavage is superficial cleavage. Uh, you might have learnt that insect eggs, they are centrolecithal eggs where a thin cytoplasm surrounds the egg. When cleavage takes place in this centrolecithal eggs, that is known as superficial cleavage because it takes place only on top of. So these are the different types. Holoblastic cleavage, where equal holoblastic cleavage which takes place in isolecithal egg, and the amount of yolk distributed, that is evenly distributed, hence from animal pole to vegetal pole division takes place. Equal cells, blastomeres, are formed, and this forms a blastula of such a type where you have a central large blastocele. This is unequal holoblastic cleavage where the blastomeres are of unequal size because it is mesolecithal egg. At some point you have the yolk distributed, so there the cells become larger in size because it takes time to divide within the yolk content, and this takes place in frog where the blastocele, you can see the blastocele displaced to one side. And this is discoidal cleavage where it takes place only on top. See here you have, so blastocele is somewhat like a slit like thing. And this is centrolecithal egg where it is superficially taking place, so division takes place in this outer or the superficial layer. Cleavage patterns. Early cleavage patterns vary widely between different groups of animals, and it's based largely on the orientation of the division planes. The simplest pattern is radial cleavage, in which successful division planes are at 90-degree angles relative to each other. The result in the blastomeres aligned directly over or side to one another. And in spiral cleavage, the division plates are not at 90-degree angles. So the blastomeres are not aligned directly over or beside one. And see radial cleavage. This is how radial cleavage takes place. Resulting daughter cells are located exactly on top of one another. They are located exactly on top of one another, and this is characteristic of deuterostomes and results in indeterminate cells where if it breaks into two organisms are, or two embryos are formed. Spiral cleavage, it occurs such that the resulting daughter cells are not located exactly on top of one another. They are located at a slight angle. Spiral cleavage is a characteristic of protostomes and results in determinant cells. Determinant cells are programmed to become specific type of cells early on during the process. So this is how spiral cleavage takes place. They are not on top of the other, but they are slightly at a different angle. Bilateral cleavage. The first cleavage results in bisection of the zygote into left and right halves. The following cleavage planes are centered on the axis and result in two halves being mirror images of one another. In bilateral holoblastic cleavage, the divisions of the blastomeres are complete and rotational cleavage. Mammals display rotational cleavage. Rotational cleavage involves a normal first division along the meridional axis, giving rise to two daughter cells. The way in which this cleavage differs is that one of the daughter cells divides meridionally while the other divides equatorially, and this type of cleavage is known as rotational cleavage. And these are the four types of holoblastic cleavage that takes place: radial, spiral, bilateral and rotational. Radial take place in echinoderms, spiral that takes place in annelids, bilateral it takes place in tunicates, and rotational takes place in mammals as well as nematodes. So isolecithal egg, Then for mesolecithal egg, it is the radial, and this takes place in amphibians. Mesolecithal, it is not equal holo, it is unequal holoblastic cleavage. This equal holoblastic cleavage, that is of 4 types. So radial one, 8 cell stage, 16 cell, 32 cell. This is a spiral one, it is at different. So this is meroblastic discoidal, where you have it on top. And holoblastic, in the absence of large concentration of yolk in holoblastic eggs, the first cleavage always occurs along the vegetal animal axis of the egg. The second cleavage is perpendicular to the first. And whereas in meroblastic, in the presence of large amount of yolk in the fertilized egg cell, the cell can undergo partial or meroblastic cleavage, where it is limited to the cytoplasm, which is